What's a dPCR machine's favorite song?
What?
Another one counts the dust. Makes no sense, but that's okay. Some of these jokes, man. ChatGPT needs a joke plugin. What did the scientists say when dPCR perfectly quantified the target? That's what I call competence.
Welcome to Absolute Gene-ius, a Thermo Fisher scientific podcast series. I'm Christina Bowens.
And I'm Jordan Ruggieri. And when they said that good things come in threes, they were totally right. We are back for season 3 of our show, and it is going to be absolutely incredible. If you're a longtime listener, welcome back. And if you're just finding our show, thanks so much for joining us. We've got a fabulous lineup of guests for you to learn from over the coming months. And we're kicking things off today with none other than Dr. Dustin Rubinstein.
Dustin is currently the director of the Advanced Genome Editing Laboratory at the University of Wisconsin-Madison. He's an expert on all things CRISPR, a researcher for new disease therapies, and a lover of dPCR, which of course we're excited to discuss today. We hope you enjoy our conversation with one of our earliest absolute cures.
When dPCR and CRISPR team up, it's a cut above the rest.
I like that one.
Well, Dustin, thank you so much for joining us today on our episode of Absolute Gene-ius. We're thrilled to have you here. Are you able to give a little bit of background, who you are, where you're at, what you're doing?
Sure. So, Dustin Rubinstein, I'm here at the University of Wisconsin. I'm at the Biotechnology Center. I lead a CRISPR-focused lab here at the Biotech Center. So we really try and provide access to all kinds of genome editing tools to whoever might need it, collaborate with a bunch of different labs, and work on projects that kind of span the mostly biomedical realm, basic science biomedical realm. But yeah, so that's me, and that's what I'm doing these days.
Awesome. So I have a couple first questions, and I know one of the first things we want to talk about is like, what does a day look like in the life of your lab, and your life look like in the day of the core lab?
I think the thing that I love about my job is I get to really interact with different scientists in different groups, right? So like I'm kind of apparently have the attention level suitable for being in this job, which is to have about 30 different projects rolling at once, right? So I'll be talking to someone about their diabetes project, and then switch over to oncology, and then switch over to neurodegeneration or things like that. So I really like interacting with people in different groups and kind of different disciplines, and I think that's really fun. That's probably one of what I spend a lot of time doing. But there's a lot of administrative stuff too, right? We've got to make sure, essentially keep the lights on, so to speak, in the science lab, but, make sure that we're, that the things that we think are funding us, and make sure that projects are moving along and, kind of helping the ship stay steady.
You don't need lights. Just as long as there's electricity for qPCR and dPCR machines, you're good, right?
Yeah, exactly. Yeah, exactly. We would put another log in the fire and get it up to 95 and then 95 degrees.
As director of the Advanced Genome Editing Laboratory, what are some of the most exciting projects or breakthroughs that you and your team might have been working on using CRISPR?
I think one of the fun things, probably one of the bigger projects that we've had throughout my time here, is developing models for neurofibromatosis type 1, which is a rare monogenetic disorder. So there's a single gene that causes it. It's rare in that it's like 1 in 3,000 people. But if you think of 3,000 people, that's not that many, and one of them probably has it. In a nutshell, we built some genome-edited pig models to recapitulate the mutations that we see in patient kids. And then, used all kinds of genomic tools, imaging tools to try and better understand the disease. That's been a really exciting project. We've got a number of other projects too. We've just developed, I guess I'm just naming pigs here, but we do more than pig stuff. But we created a pig that we call Onco-pig, that we can actually induce genes that induce cancer. So if you're a drug company and you think you might have a new drug for kidney cancer, we could give them kidney cancer. And if you think it might work well for, liver cancer, we could give the pig liver cancer and induce cancers that way.
Can you talk a little bit about why are pig models beneficial for things like cancer research? Why not other models like zebrafish or mouse? Or, is there a particular reason that pig models are useful?
That's a great question. And it's funny because we're just coming hot off the heels of the International Biomedical Swine Conference that just wrapped up. So I think it kind of gets back to the whole idea of tools complementing each other, right? No one's ever using digital PCR, for all aspects, and no one's ever doing all DNA sequencing. Every tool's got its right place. We've learned a lot about basic biology and some translational biology from invertebrate models like Drosophila and yeast from Danio and zebrafish, and we're learning a lot from mice and rats, but they don't do everything well, and probably some good evidence for that is how extraordinarily expensive it is to bring a drug into the clinic, and then how overwhelmingly likely it is to fail. So, you spend what seems like a whole lot of money developing the idea for a drug and designing and making the drug and checking that the drug works in small animal models, but that's nothing compared to actually bringing it into the clinic and giving it to humans. And then after that is when it usually fails. So why does it fail? I think that's probably a relevant question that we're all kind of asking each other. And some of that is because, well, maybe the models that we were using weren't really the best models that we had on hand. Like I said, we've learned a lot from all kinds of models. Like I don't, there is no one model. We need all kinds of different models to ask different questions in the right context. But, if we're thinking about cancer, we can study mice and study like cancer in mice. But generally, when mice get cancer, experiments with mice are usually measured on, months to weeks. Whereas cancer for humans, we often, one of the buzzwords you hear when you talk about cancer in humans is management, right? It's not just about like cure the cancer, right? Like, oh, I got cancer, I'll take a pill and now I'm cured. My headache's gone and my cancer's cured, right? I mean, it's about management, right? It's a long-term process about maybe like a, first step. How do we manage what's the first thing we need to do to try and knock back the cancer? And then how do we track it and see how well that worked? And then how about, circle back and do, another dose or something. The problem is that mice generally get cancer and die. Whereas, with humans, we're talking about, managing this problem that is cancer. With mice, they get cancer and then they die. So that is not really a great model for the way cancer actually works in humans. Pigs have a much similar developmental trajectory to humans. They're much more similar in size, right? So I'm like from the suburbs. So I always think when I heard pigs, I always thought like Charlotte's Web, like like cute little pigs. But I didn't realize that pigs are basically like horses with short legs. Like they're enormous. So if you want a pig that's actually going to match a human size, you have to use what are called mini pigs. So when I'm talking about biomedical pigs, it's often these mini pig varieties that, again, are not that many. They're not like the cute ones you carry in your purse, right? That, they're super adorable. They're 150 pounds ish, or a little higher. The idea is that they match humans.
That's awesome. So Dustin, you and I have known each other now for quite a number of years at this point. You were one of the early adopters of the Absolute Gene itself, but I know that you've been using dPCR for quite a long time. Can you describe, why your lab decided to, integrate dPCR technology in, and kind of the reason why you find it to be such an important technology to have in your lab as a tool for the genome editing?
Yeah, I think what really hooked us was just how easy and precise it was to get an answer for things that like, maybe qPCR, which is great, like in some respects, but, sort of the reliability and the precision that you get from digital PCR was really helped us hurry things along. Like one of the big applications that we had were copy number variant assays, and not really maybe in the way that most people, for all you real qPCR folks out there, maybe a little different in the way that you might usually think of it, but more in terms to confirm the edits that we made, and that's where digital PCR became like really a really critical component for us as sort of kind of a QC quality assurance step.
Can you talk a little bit more about like why that works in your favor for digital PCR? Because I know that's a topic that comes up a lot, and I know our listeners might like to have a little bit of education about why digital PCR is advantageous for kind of understanding gene editing confirmation.
In a nutshell, I think it basically comes down to this. So when you're doing genome editing, you make the change, and then obviously it's really important to know what the change you just made is, right? That becomes the next big problem. What's the change you just made? Almost everyone uses some sort of PCR-based method to read that out. That PCR-based methods can sometimes like kind of add like a little fog. It can help you kind of maybe see what you want to see rather than what's actually there. So you might be trying to amplify a certain part of the genome, and you get a PCR band, and you think, okay, great. This fragment exists in the genome just as I think it did. But one example might be that, well, maybe that fragment exists in the genome, but only because maybe there are many, many copies of that insert, and that fragment exists as well as tons of other fragments that you can't see with PCR, right? And the other thing is sometimes you PCR something and we might forget that we actually have like 2 chromosomes, and you're not just PCRing and getting a band from one. You'd want to make sure that you're interrogating both chromosomes. But when you do an endpoint PCR, you have no idea like which chromosome gave you that band, right? So using digital PCR kind of allows you to tease out some of those problems and address them. So we're finding that these are kind of really key things to do. And it turns out when you actually like peel back like the layers, sometimes it's not as pretty as you think you did, as you think it is.
Yeah, so it gives you a little bit more precision as opposed to some of the other methods that have been around for a little bit more time and, maybe don't have quite the resolution.
Yeah, exactly. Right. Exactly. And it like allows us to test those, and then it does it in a manner that's like so much more precise and really, really fast and easy. So what's not to like there?
Yeah, Justin, I actually want to take even one step back and talk a little bit about genome editing and CRISPR technology. Is that something you can comment on? I know it's a newer technology. I think it's definitely getting to be more well known and more well utilized in labs. But what does CRISPR actually allow you to do when it comes to gene editing techniques in your lab?
Yeah, boy, yeah, that's hard. It's really exciting. So, I think from a fundamental perspective, we've always thought of genomes. It would be described in terms of like hard copies, right? It would be like if the human genome was printed, it would be like volumes that would go from the floor to the sea, because we always talked about reading the genome and what's the genome sequence. But, for the first time, we actually had the ability to not just, read it in a printed form, but to actually like open it up in Word and change it, rather than kind of making really passive inferences about like, well, these patients have this mutation and these patients don't. So therefore it, correlates pretty well with like, a disease. Of course, correlation is not causation. What you need to do in any science experiment is to actually make the manipulation. And that's what CRISPR really finally allowed us to do. So that's kind of the really exciting thing, is finally get a causality of genomics, which is kind of huge. But things are moving so fast. That's sort of like CRISPR version 1, right? Now, one of the concerns with CRISPR was that, well, you create this double-stranded break, which means, you create a break in the DNA. When you create a break in the DNA, the cell freaks out, because if it can't fix that, it's mitotically dead, right? You can't just have like, chromosomal arm floating away. So there are many, many DNA repair pathways that are always monitoring the safety and the health of, the chromosomal arms. And sometimes, it might, those mechanisms might try and stitch the ends back together in ways that like you don't really want, right? Or maybe something really funky could be happening that might like cause more problems than you wished. So there are actually versions of CRISPR now where you don't even have to cut the DNA, right? Whether it's base editing or prime editing, some of these other really exciting things where, you're just using CRISPR as a homing device into like a certain spot in the genome. And then once it's there, you can stick whatever kind of funky tools you want on it. There's all kinds of really creative ideas, and some of them are working, which is great.
So I have kind of a question to think about how your lab assesses new techniques and technologies to bring on board to keep up to date and keep up to standard. So how does that go about? How do you make sure that your lab is keeping up to date? How do you decide what to bring on when it's the right time? And how do people who are collaborating with your groups get to leverage these technologies? Is it just through the products that you're developing? You mentioned you're developing these models that the groups can kind of work with, or is it more in a service base? Can you talk a little bit about how all that works?
Yeah. No, I think that's a great question, because trying to stay in the know with CRISPR is sort of like sipping from a fire hose, because like every time you like check, there's like a new acronym with like CRISPR tagged onto it, and it's like a tiny variation of the thing before, right? So at what point is it something that you really need to pay attention to? For us, I think it's really like, keep it simple. What is it that you need, right? Are your needs being addressed with this platform as it is? And if not, are your needs like so much more advanced that you're really willing to put in the cost to try this other thing? I think it kind of comes down to something like that. We really get by in the fact that we're experts in kind of one set of tools. People generally they come to us because we sort of know these tools. We know what's reasonable to try and what's like just not a good fit. We work with them on a kind of a case-by-case basis and see, try and find the toolkit that works with them.
Do you ever come across cases where you may actually have to utilize multiple different technologies? Maybe it's digital PCR and sequencing, or digital PCR and qPCR, in order to solve a problem.
Yeah, I mean, you're exactly right. I mean, digital PCR is important, but just like no one person can do everything by their own, no one tool is going to fix everything, right? So just to give you like a broad sense, like we create these models, right? And we want to make sure that at the end of the day, we've got a model that we know exactly what it is, right? We can like sequence confirm and say that's exactly right. So we can start with like the PCR and we'll know which ones we can throw away, right? And we'll throw out a whole bunch of ones that aren't so good. The next step we want to do is use like, digital PCR and ask, okay, like of those, can we get rid of the ones that look good by PCR, but maybe weren't quite right? And then we, throw out a whole bunch of other ones. And then, our new standard now is to go to long read whole genome sequencing and confirm that way, right? So then we'll pick, basically like one allele and sequence that and be like, okay, is this exactly what we think it is? These tools don't exist in a vacuum, and they're all really complementary. The shortcomings of one can complement the strengths of another.
How are you, designing your assays, for digital PCR? Is there any particular assay or types of assays that you utilize frequently?
Yeah, so like I said, we probably do more CNV analysis than anything else, right? So what does that mean? So we're generally targeting somewhere in the place we're trying to edit, right? Like a representative tiny little chunk in maybe whatever let's say we humanize a gene and we're going to target part of that humanized little gene. So that'll be one assay, and then we'll run it in duplex with some other random spot in the genome, right? Something that we haven't edited, most likely. It would be really bad luck if we did, but something that should still have 2 copies, right? Or maybe it's a cell line that has 3 of those chromosomes or something. But, so now we know exactly how many copies of the gene, because we measured that other random spot, and we know how many, down to like the molecule, which is kind of amazing thing about digital PCR, right? Is it gives you an absolute number. So we know exactly how many copies of that other random spot is there. So therefore that means we know how many copies of the genome are in our sample. So then we can say, well, if we have this many copies of the genome in the sample, how many copies of our humanized gene do we have? And that's kind of the strategy of sort of what we often do.
Can you talk, because I know a lot of this you'd be designing kind of custom targets for these specific edits that you're making. Can you talk a little bit about what you do for like optimizing assays? Do you find yourself having to optimize a lot of assays? Do you find yourself using different platforms? I know a lot of users find themselves kind of optimizing on qPCR and then moving to dPCR. Do you have any strategies that your lab uses to do that?
That is like a evergreen question in our lab. Sometimes we have issues with optimizing, and we're kind of like, it's always that question, like, is it worth like spending time optimizing it, or should we just kind of move ahead with what we have? So we've been lucky. I mean, Thermo Fisher actually has been pretty kind to us about helping us and guiding us with some of those assays. But yeah, generally we take it right out of the box and try and let it rip. And if that doesn't work, then we'll go back and see what we need to do.
Are there any types of features of like the digital PCR systems that were really important for you when looking for systems that are that were important when you're integrating a system into your lab?
Yes. Number one, we wanted it to be something easy. I mean, there are all kinds of different ways to make PCR digital, and just the simplest and quickest way we could get those preps done, the better. So that was probably one of our first interests. I think the few other things that were really important, of course, it had to be reliable and had to be precise, obviously, or else we're not going to use it. But for us, like, we don't always necessarily have the same type of like sample input, right? It's not like we always just have like DNA that's like as pure as the driven snow. Sometimes we take lazy shortcuts. They're not lazy, but just we don't have that much input, so they're kind of, like cruder representation. So we also needed a system that worked on all kinds of different levels of they need to be tolerant to all kinds of other potential contaminants in the sample. And for us, once we screened a few different platforms, it was pretty clear which one was most robust for us.
Talking a little bit more about, CRISPR, in your opinion, what are the most promising applications of CRISPR technology in maybe understanding mechanisms behind disease genetics and research behind the development of novel therapies?
I think one of the things that's really exciting about thinking about using CRISPR to cure diseases is that you might think, well, using CRISPR to cure disease, you just change the mutation, right? That is causing the disease. I mean, that'd be great if it was that easy, but sometimes it's not. So a lot of times what it does is it forces you to go back to the basic biology, right? So to understand what are the genetic networks. So there might be a particular mutation that's causing a disease, but like, what is the genetic network in which that mutation is causing the disease? Because now all of a sudden, now you're not tethered to just that mutation. Now you can find all these other potential genes in the network. So if you look at a kind of eye-poppingly success story of sickle cell, right? I mean, there is a single mutation that segregates in the population, right? I mean, there's one nucleotide that changes that is responsible for sickle cell. And using CRISPR to cure sickle cell the goal wasn't to fix that mutation. It was, well, what are the other genes that we could maybe hit to actually find another way to cure it? And so really that's what they did. It was basically an understanding of how that genetic network happens throughout the course of, human lifetime. So it really kind of shows that there's always going to be a place for basic biology, right? It's never going to be as simple as just changing a mutation that was discovered. But also, some crazy things that look like science fiction now too, right? So I don't know how much, you've heard about xenotransplantation, right? Before I started applying CRISPR tools to the idea of organ transplants, it was shocking to me to learn that you're more likely to die waiting for an organ transplant than you are to receive it. I mean, I realized like how really acute this issue is. And there had been a long-standing interest in maybe looking to the pig, historically pre-CRISPR, and thinking maybe about, looking to the pig and asking, well, okay, so there's an animal that's, kind of human-sized. It's a mammal. It's got similar developmental, timelines. Maybe we could kind of like, it kind of would fit about in that spot maybe. But the thought was always, well, how can we change the human immune system to accommodate a pig? For many, many years, that was kind of the thought. Like, how can we make humans accommodate pigs? And I'm sure you'll see where this is going. But now the idea is, how can we change the genome of pigs so that they'll actually fit in nicely to a human, right? So now we can ask, how can we make a pig accommodate the human immune system, which is one of the kind of really exciting things that's happening. And there's been some tremendous steps forward and progress in that. We have a Xeno program here that we're really excited and proud of. Some of these benefits seem to almost be like science fiction, right? But here they are, I guess.
Are you looking to precisely quantify targets with digital PCR without a standard curve, measure viral titer, or quantitate residual DNA? Are you looking to scale your dPCR workflow while maintaining a simple and efficient process? If so, Applied Biosystems QuantStudio Absolute Q Auto-Run dPCR Suite is for you.
The QuantStudio Absolute Q Auto-Run dPCR Suite is an automated, easy-to-use dPCR solution that helps improve throughput and utilization for absolute quantification of your samples. The system enables efficient utilization with multiple users able to load and schedule runs, along with greater productivity with automated plate loading and run scheduling with a single click.
It also enables consistency and multi-plate analysis so you can analyze batch sizes that match your experimental needs together for improved reproducibility.
You can learn more at www.thermofisher.com/autorun. That's www.thermofisher.com/autorun. The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
And now back to our conversation.
So, Dustin, welcome to the career corner element of the podcast. In this corner, we like to talk a little bit about how you got to where you are and kind of your career pathway. Could you give us a little bit of background on your educational background? What kind of training did you receive, and maybe even a little bit of how you got to where you are today?
Yeah. All right. So I went to undergrad at the University of Illinois in Champaign-Urbana. Moved to Italy, New York. Got my PhD in neurobiology and behavior at Cornell University. Maybe that kind of like started a seed, because that department is a little unique in that in the same department we had people who were going out to like islands off the coast of Colombia and studying bird song and ziplining off canopies and trying to understand the function of bird song, all the way down to people who are understanding how individual genes and individual neurons are affecting the way nervous systems grow up and develop, and trying to cover that ground in between. And maybe that's why I can't really sit down and focus on one topic. And and I love all kinds of fields of biology. That was sort of my home department as a grad student. I was always interested in sort of the genetic basis of behavior, right? So how can genes influence the nervous system to be set up so that we engage in these, crazy complex behavioral patterns? So I studied Drosophila and Drosophila courtship song. So males actually sing to females, and the song has to be just right. But somehow that's not learned, right? It's programmed into their nervous system. And for the males to make it and for the females to prefer a certain type. I mean, that's pretty cool. So I was kind of a behavioral genetics person and understanding how the nervous system changes. So I was in a lab studying neural development and understanding the genes that, affect synapses and synapse growth and synaptic changes. And that's the time when those CRISPR papers finally hit the airways, right? When totally changed the way geneticists, did their work. So we worked with some amazing folks in a group with several different labs on campus, and we got CRISPR up and running in Drosophila. Around that time, if you knew what CRISPR was, let alone, you actually got it working, people were, bothering you all the time to figure out how you can help them. And realized that I could kind of take this background that I have and maybe help others. And that's when I was asked to found this CRISPR core here. So I was basically formally asked to do that, right? Actually get paid to help other people with their CRISPR projects. And that's kind of what landed me in this seat here. And here I am talking to you.
What a journey. You mentioned even the bioinformatics with the website. How has maybe taking part in different opportunities shaped the skill set that you have now? Have you noticed that being part of multiple different things has kind of helped with your core lab and helping run your lab now?
Yeah, I think it kind of goes back to that. It goes back to that idea that we're all we have to surround ourselves by experts in tons of different fields. I wasn't necessarily a pure bioinformatics person, but I was lucky enough to be able to work with them in close proximity here at the Biotech Center. I'm still not really officially a cancer biologist, but I'm lucky to have the UW Comprehensive Cancer Center here, layering in different forms of expertise, which is also kind of layering different people in different backgrounds, has really enhanced the way, I think about problems and try and find solutions for them.
I think this segues perfectly into my next question is mentors. A common theme that we've seen on a lot of the with a lot of podcast guests is that they had mentors that kind of helped guide them and helped shape their knowledge and skill sets. Is that also true on your end? Did you have any mentors that really helped build your skill set and knowledge to get you to where you are today?
Absolutely. I wouldn't be where I was today if it wasn't for, a bunch of amazing mentors that I've had. I'd have to say, I wouldn't be where I was now if it wasn't for sort of the bravery and audacity of my advisor, Kate O'Connor-Giles, who I mentioned before. Just a brilliant person who was sort of fearless and just knew if we just kind of keep doing the work we need to do. We'll get this, project done and wrapped up, and then, kind of can get back to our regularly scheduled synaptic development experiments. Having her teaching me to see, to have that, vision to follow through, but finding a place for me in it as well. I wouldn't be where I was now if it wasn't for her.
Absolutely amazing. How about anything, one or two things, whatever comes to mind, that you have learned in your career that really sticks out, that you think would benefit others that are maybe early in their career, or even later in their career, and any tidbits or things that you've learned in your pathway?
I think it's primarily that you can never really plan your future, right? I mean, you want to have a goal, you know where you want to go, and you take the steps to get there. But there will be other opportunities, there'll be other doors that will open, and you need to be ready for those things that may not be, exactly the same destination you're going, but you want to at least be open-minded enough to think, well, this is a pretty cool opportunity that I just hadn't really considered. So I think don't necessarily get wrapped up so much in this ideal endpoint that you think you need to be in, but look around you, and there are probably opportunities, if you just kind of take a moment and realize what your role is in this situation right now. There are probably opportunities that are different than what you thought, but something that really plays into things you love and things you're good at. Kind of just have an open mind and be willing to take take what life hands you, I guess.
I have one more. It's a two-parter. One more question that we ask everybody: what is your most embarrassing lab moment? And then on the flip side, what are you the proudest of?
I'm trying to think through all the explosions and the egregious pipetting errors.
It can be a general thing too, Dustin. It doesn't have to be one story.
I'm trying to think of a time I tripped and fell. I know, I got to give you something. I spilled coffee on myself. I know. I don't know. I'm so bad at these questions. My proudest. I don't know. I'm kind of proud of this group that we have now. I think we all bring some really, exciting, unique talents, and I think we're doing some really cool stuff. And I'm excited for what's in store. I don't know. I mean, it sounds like a cop out.
That's a good answer from a director, right?
Right. It's better than the alternative. Well,
I've got 8 ball and chains hanging on my leg.
Really proud of things long gone, the good old days. You have no idea.
Christina, any questions on your end? I am career questioned out here.
No, I think that's it. It's been so nice having you on the podcast, and really nice talking to you again. It's been absolutely great.
Christina, it's always great. It's good to see you again. Jordan, it was super fun chatting with you.
That was Dr. Dustin Rubinstein, director of the Advanced Genome Editing Laboratory at the University of Wisconsin-Madison. With more fascinating conversations around the corner in upcoming Season 3 episodes, subscribe now and get notified when new episodes drop. Stay curious, and we'll see you next time. This episode of Absolute Gene-ius was produced by Sarah Briganti, Matt Ferris, and Matthew Stock. Jordan, why don't skeletons fight each other?
Oh, why don't skeletons fight each other?
They don't have the guts. That would be a good Halloween one.
That would be.